Hello everyone, this is Yuan. You can also call me Ken. Currently, I'm enrolled in UCL Chemical Research Program and Joint Metpolar Group, where I'm aiming to understand the chemical building blocks of life by exploring peptide ligation in a prebiotic environment. And I'm going to present to you what I understand to be the chemical prospect on the origin of life. Finally, I will briefly introduce my own research content as well. Thank you. Hello everyone, today I will introduce prebiotic chemistry. The origin of life is a continuing scientific discussion and research. This is a field that brings together biology, history, chemistry, and geography, and can date back to 4 billion years ago. Prebiotic chemistry is the study of how life might have arisen on early Earth through chemical reactions. Something like ammonia, carbon dioxide, and water in the atmosphere, together with metals in rocks or some other natural occurring substances on the Earth, were used by nature to produce precursors to protons, genetic carriers, and lipids, which are essential for life. Understanding the chemical processes that lead to the emergence of life on Earth is indeed a fundamental problem that requires the expertise of many different fields, including organic chemistry. The Earth just formed about 4.5 billion years ago, and it has undergone many changes since its formation. In the early stages of its history, the Earth was a very hot ball of rock and metal, with no surface and a thin atmosphere of gas. Over the course of its history, the Earth was undergone many major events and changes that have shaped the planet and its environment. This includes the emergence of life, the formation of continents, and the development of atmosphere and climate. So, all of the life on the Earth today is all related in a very close sense. That all have the same sense. They all have the same genetic information and all the ways of processing genetic information. But is the origin inevitable? Is it inevitable that we would always end up with the same type of life? So, how do we address this question? There are lots of different ways, and labs around the world address this question in lots of different ways. Maybe the first way that jump out, and this is a common way to think about this question, is imagine the environment that would lead to life. We could have lightning or submarine or drying lagoons, atmosphere aerosols, big impact craters that then lead to chemistry from that impact. And then we can evaluate the types of chemistry that occur there and say if that lead to life. But the problem with this approach is there's really insufficient information. When we get back to the early origins of life and we get back to the Hampton period of geochemistry, we really have so few rocks to available and analyze. So later data in these rocks that there is not information to constrain the types of. And before we think about the source of the life in other ways or said in chemical ways, I want to give you a sense of the three building blocks of life. These three primary building blocks of life are nucleic acids, protons, and lipids. These molecules are essential for the structure, function, and regulation of living organisms. First, just the nucleic acid. Nucleic acids are biopolymers made up of nucleotides monomers. There are two types of nucleic acid, DNA and RNA, this we all know, and nucleic acids play a vertical role in the storage, transmission, and expression of genetic information. Proteins are large, complex molecules composed of amino acid monomers. They are involved in vertically various aspects of cellular structures and functions. Proteins serve as enzyme catalysts that facilitate chemical reactions within cells. They are essential for the growth, repair, and maybe regulation of tissues and and the last one, the lipids. It plays a fundamental role in the structure and function of biological membrane, And maybe I don't think there's a lot of discovery in this period. It's also in its process. Now that we have talked about the building blocks of life, the next thing we need to think about is how to do it chemically. Of course, this would have to be operating a prebiotic environment. And my personal research topic is to building peptide ligation systems that can self-accessible process information, transport, and control reactions within this network. A peptide is a chain with amino acids linked by peptide bonds. Peptides are also considered to be the building blocks of protons, monomeric units that make up protons as one of the three building blocks of life, along with RNA and lipids or DNA. And it also plays a very important role in cell metabolism and growth. In 2019, our lab published a study on peptide ligation by chemi-selective amino-nitride coupling in water. Before that, there is no chemical methods of achieving alpha peptide ligation in water that tolerates all of the 20 amino acids that the peptide ligation. As the project progressed, it became clear that soy ester played a critical role throughout the process. It can provide the high selectivity required for ligation, and my experiment will be based on this experience. First, book group would protect the amino end of the different amino acids with different side groups, and then ethanol will be added to the acid side and make it have a very good living group. And of course, the most important thing, which I'm actually doing now and will continue to do for some time to come, is to see if these different amino acids in water and under different pH conditions can connect to each other smoothly, or if there is a loop or something that blocks the ideal state. And at the same time, we also know that glycine is one of the most common amino acid and one of the simplest. I also want to understand the reactions of different amino acids with glycine. So this was something that had never been studied and published before, and it should be a very long process and difficult process. That's all, and thank you very much, and I hope you will keep following our progress. For more information, please click on the link of Pono Group website below, and thank you again for everyone's attention. Thank you.